Hello, today we're doing cell cycle, mitosis, binary fission, and meiosis. I hope you enjoy. So cell cycle, there's interphase and mitosis. So for interphase, it's most of the cell cycle basically, like 90% of it. There's the G1 phase, first gap phase, think growth. So the cell grows, makes organelles and molecules. S phase, think setup. So DNA copies, centrosomes copied, or an analogous process in plant cells. G2 phase, second gap phase, grows, makes organelles and molecules. Alright, so now we'll go over mitosis. It's so we have prophase, metaphase, anaphase, and telophase. And you can, some people split prophase into early and late prophase. I don't think I'm going to do that. So for prophase, the mitotic spindle forms chromosomes condense, and nuclei disappear. Okay. Metaphase, chromosomes align at something called the metaphase plate. Ready to have something called sister chromatids, which are copies of the chromosome to have sister chromatids separated. In anaphase, the chromosomes are pulled apart and telophase is basically the inverse of prophase. So nuclei reform in the two new cells. Chromosomes decondense, and the mitotic spindle disappears. Okay, so now we'll go over binary fission in bacteria. So basically the circular chromosome of the bacteria copied, pulled apart, then bacteria is split. Also, something we forgot in mitosis, there's something called cytokinesis. Could happen during anaphase or telophase. Basically splits the cell apart. That's important. So now finally, we'll cover meiosis. Meiosis used to make cells that are important for you to reproduce, I guess. So we have meiosis 1 and meiosis 2. And so in meiosis 1, first we have something called crossing over of homolog pairs. So when they say you have 23 pairs of chromosomes, this is sort of what they're talking about. So if you have chromosomes that look like this, you can cross over like trade pairs like this, as you can see. So that happens. And then after that, it's basically mitosis, but separating homolog pairs instead of sister chromatids. Instead of sister chromatids. Okay, and then we have meiosis 2. So we have 2 cells left over from meiosis 1. They go right into meiosis 2, which is basically mitosis for these haploid, for these haploid cells, 4 haploid cells. And what does that mean? So you have your chromosome still looks like this, and you basically need to split that apart, and that's pretty much it. Okay, so I hope you enjoyed.